program head of the Environmental Microbiology program, as well as the Molecular and Microbial Genomics program at the BC Centre for Disease Control. A really good example of where Canadians have had some leadership in using technologies to deal with this outbreak is the field of genomics. So genomics is the perfect example of an emerging technology that was helping with this outbreak specifically. If we look at SARS in 2003, it was about 6 months to generate a complete genome of that virus. Here with this outbreak, it was about 2 weeks. So the WHO first became aware of a pneumonia of unknown origin at the end of December. And on January 11th, the first genome was publicly released. And this was really, really important for a few reasons. The first is that it allowed us to develop diagnostic tools or tests to be able to know if somebody had COVID or not. But it also set the precedent for using genomics to study this outbreak and to release that data publicly quickly. And so we're using that technology to understand how the disease is spreading here in Canada and how it spreads here in British Columbia. And that could not have been done without emerging technologies such as next generation sequencing. And I can really speak more about the testing technologies as opposed to the informatics technologies, but people have been talking about machine learning, AI, and being able to crunch the data. And I think that's one of the things that's coming out in this outbreak is just how much data is available and how quickly. And that's quite different than in previous outbreaks. And so whether or not it's an entire genome of a virus or the number of tests that are performed or creating these projected models, we're living in a data-rich environment. And so those more advanced tools are certainly needed. So there's a number of different laboratory tools that are proving to be important in this outbreak. So the initial molecular-based or RNA-based diagnostic tools, the genomics to understand the spread. We do expect that understanding people's antibody responses is going to be quite important as well. And so that's the serology that you might hear people talking about. So these are all different laboratory tools that will be really important. And I think that one of the things that continues to be important is how quickly these tests are being developed and deployed. And that, for this outbreak and future outbreaks, development and deployment are really important factors in preparedness. Genome sequencing is basically sequencing the entire genetic code of a microorganism. So in this case, we would sequence all the RNA that makes COVID. The reason that we're doing this is for a few reasons. In the very beginning, it was used to actually ascertain what was this organism that was causing this unknown pneumonia. After that, it was used to identify diagnostic methods to detect this organism. So we sequence a number of different genomes and determine where there are stable sequences that we can actually use for diagnosis. And now we're sort of at the point where we're using those genomic sequences to understand transmission. Does case A and case B, which may not be related, if they have identical sequences, what does that mean? Or for example, in our scenario, in the very beginning of the outbreak, we could really link people's travel history to their the genetic sequence of the virus that they were carrying. And it really helped us understand how the virus was spreading and where it was coming from. The other tools, the genomics can provide so many different types of data. It can help us understand how a vaccine might work, whether or not we might expect to have vaccine boosters or new strains or new vaccines every year. And what we're seeing with this virus so far is that it's pretty stable. It doesn't change a lot. We don't expect recombination like we do with influenza. And so it might be that a vaccine that's produced might provide protection for quite some time. And we can learn that from the genome. We can predict that from the genome sequences. Likewise, we can understand what some of the therapeutic interventions might be, what types of drugs might work best against this virus, just based on the genetic sequence. So it helps us in many realms of understanding the disease and the impacts it has on the patients. Everything is COVID right now, and that really is our priority. The diagnostics, the research, everything is focused on COVID, and that's because that is our main priority. Our main priority is to fight this disease. in general, what I was doing prior to COVID-19 was really focusing on the operationalization of genomics. So when we're talking about genomics, that's sequencing the entire genome of a bacteria or a virus. And in this case, we've been using genomics for a while to study foodborne illnesses, which I'm very interested in. And so being able to find the genetic sequence of a pathogen in a human and in the food that they consume and matching them and saying, yes, in fact, this is where they became sick. And that really helps us in how we investigate outbreaks and what measures can be put into place to prevent future outbreaks. We're doing similar things for COVID-19 to be able to use that sequence of the entire genome to understand whether or not two cases are related to one another. And so that is a really, really important technology, and it's actually going to transform the way we do microbiology. But of course, there's a big learning curve for that, and we're trying to really find the way to best implement it. It's expensive, it requires new training, new technologies, but we really want to be able to understand how we can transform the way we do public health in the future. For me, this is the first pandemic I've been involved in. So I think but there's certainly a history of different epidemics and pandemics that have affected Canada. So there was Ebola, which didn't actually come to Canada, but we were very working on the preparedness for that. We had H1N1 in 2009. And then we had SARS in 2003. And I think that some of these previous pandemics really shaped the way that we respond to public health emergencies, and also how we integrate the research community, because it's more than just the frontline physicians and the frontline public health workers that need to be involved in this. There is certainly a research domain that can help accelerate our response. So I would say that Canada has certainly learned from those previous experiences. And I think that what you see with these research calls from Genome BC, Genome Canada, CAHR is that people really want to invest. The funders really want to invest in the research that's being done, and that can happen in a really fast pace. And by making that funding available quickly, that means that we can accelerate the work that can happen. So the turnaround times for some of these funding opportunities is sometimes less than a week. And I think that quick pace really allows the quick pace of research. And you'll see that, if you look at the CHR website or Genome BC website, you'll see there's a lot of different types of funding that's been funded, or research that's been funded here in Canada. Everything from therapeutics to genomics to vaccine development to just modeling how the disease is progressing, or even understanding the social impacts of this disease. And I think that the breadth and the number of researchers that have been funded really shows Canada's research capacity and the investment from our government and our funding agencies to support that. Collaboration is really key for fighting this disease, and I think that you'll see that happening at multiple scales, whether or not it's at the level of trade, sharing PPE, being able to work together in this crisis. I'll give you a really good example of how collaboration has occurred. So the first qPCR, or quantitative PCR assay that's used for diagnosis, it was first released on the World Health Organization. One researcher uploaded their method. We're now using part of that method, and that was uploaded mid-January. And I think in a previous era, somebody might have waited to publish that and get it into the peer review literature and get all the kudos for that. But that's not what's happening here. What's happening is that people are releasing their methods without going through peer review. They're making things available so that we don't have to wait 2 to 3 months to have data available or methods available. And that collaboration, we emailed those researchers and said, what if we did it this way? What if we did it that way? And we had that collaboration to be able to produce a good product really quickly. And likewise, we developed our own method, and we've shared that with different researchers across Canada and different sites within British Columbia. And I think this isn't the time for glory. This is the time for sharing and collaboration, and to help each other advance the methods. And so I think that, in terms of method development and sharing of data, that has been really, really important. And I don't think we could be where we are today with respect to our testing capacity had we not collaborated with the international community. One of the challenges with collaboration is that things are just changing so quickly. So within days, a new project has emerged, and trying to get everyone together and chatting about it can be really difficult, given that everybody has so much work to do. And I think that that's just the nature of the beast. I mean, all we can do is work on ways to communicate better. I've started using Slack, which I've never used before, to learn about what other people are doing and as a tool to communicate between researchers. There's been lots of emails of connecting one another, and we're really in that sense of just helping one another at this point. I don't know if we'll see a pandemic this big again, but there will be other emerging diseases. This won't be the last time that a new disease emerges. And so I think that one of our priorities is just making sure that we have the ability to scale up a response, just like this time. I think that we saw a really rapid response and a really aggressive response, and I think it's really is a testament to the science and the data that was available to support making those kinds of decisions. And I think that continued investment in public health and research will be important to making sure that we can have that rapid response in the future. None of this can be done without funding. And so that's one of the things I would say to be prepared for future pandemics it really does mean to invest in public health and invest in testing technologies or scientific technologies and research in general. I think that there's a lot of the innovation that we're seeing is because of past investments. And so we really need to make sure that those investments continue so that we can continue to have a strong public health function and a strong research community. Investing in academic research is really important. Continued research on like basic science as well as applied sciences. I think investment in public health and investment in training programs for public health is really important. So one of the key tools for doing some of the work that we do is genomics, but there isn't a genomic specific training program either on the laboratory side or on the informatics side. I think continued investment in informatics and modeling will be very important. One of the things that we have learned from this outbreak is that we are very dependent on technology companies outside of Canada. And so with the global demand for testing supplies, for example, we're in competition with other nations in terms of getting supplies. And so that's been one of the drivers behind some of the testing challenges is the accessibility to testing kits. And so I think that building the Canadian economy so that we can, for the health sector, we can rely on Canadian suppliers would be very important in the future. So I do think that, we are seeing a number of Canadian companies repurposing some of their production for things like personal protective equipment, and perhaps in the future for materials for diagnostic tests. So I think that may be something that we see in the future. I think a lot of attention has been placed on testing numbers, how many people have been tested, but nobody's really been talking about the method to test or what we're doing with the data. And I think that we really should be thinking about where we are in terms of the automation, the testing technologies, the genomic technologies. We've come a long way. And had this outbreak happened 10 years ago or even 5 years ago, we would not be in the place that we are today. And I think that that's something we should be really focusing on is the technology side of things and how, how grateful we should be that how far along we've progressed over time, and then the investments that are needed to continue to improve the technology yields in this country. Today we were chatting about the fact that our respiratory illness diagnoses are down right now. Likewise, the rates of diarrheal disease is down as well. And that's not for lack of submissions. That is because we believe that social distancing and increased hygiene are contributing to a decrease in the spread of infectious diseases. And we actually saw this during H1N1 as well. So when that outbreak of influenza occurred, we saw a lot less diarrhea. And that was because changes in isolation, changes in cleaning procedures, changes in hand hygiene. People are worried about contracting the disease. And so, I've been washing my doorknobs, and my kids sing happy birthday twice, and they wash their hands many, many times a day more than they ever did before. And I think that that's actually, as a society, if we can actually move towards some of these better hygiene practices, we might see a reduction in the burden of some infectious diseases. So not that there should be a silver lining of any of this, but I do think that better knowledge of hygiene can only be a good thing for society. probably the one of the most stressful times of my life. I think that, but this is what, I mean, we're in public health for a reason, and we're there for this type of scenario. And I think this is what really motivates us. And so I think that there's, of course, there's a lot of pressure, but that is our job. And I think that we're all, as people working in public health, working in research, this is sort of what we're built for. And I think that we're happy to rise to the cause. And that's not to say that there hasn't been many sleepless nights, but it is our responsibility. Well, I have 2 young children at home, and I think that it's been so nice to go home to them and to have the cuddles and to have that time with them. And I think that adds so much fulfillment to my life, and to, really, it's such a stress reliever to have a dance party with your 2-year-old and 5-year-old. I think that's, that's the way that I de-stress, is just to be silly with them. I think that's been, the times to walk around the neighborhood with them and to just take that time to be with my family has been really important. I am Natalie Prystajecky, and this is My Future Economy.